almost 1 in 300 people are at risk of developing motor neuron disease over their lifetime. It's 1 in 200 for schizophrenia. About 50 million people worldwide are suffering from dementia, and these numbers are going to triple by mid-century, and we still do not have interventions that can, forget curing, even slow down the course of these disorders.
For us to build a comprehensive picture of disease mechanisms, etiology and progression, we need a multidisciplinary and multifaceted approach in order to bring together imaging markers alongside other markers such as genetic, clinical, fluid biomarkers.
The University of Exeter is really pioneering the way of understanding the biological basis of brain disorders. We have a world-leading program in epigenetic research, and in this we study the ways in which the genes that define the different cell types of the human central nervous system are expressed. We look at the ways in which those genes are turned on or off, and the ways in which these normal patterns of gene expression go wrong in the context of diseases.
Exeter has a holistic view of approaching neuroscience in terms of not in the basic biology but also in disease interventions. We have all the way from discovering what's wrong with the disease using cutting-edge models and patient tissue, as well as applying advanced genomic techniques. We also have the means to actually intervene and then even capture the disease early in the stage using advanced medical imaging techniques.
My research uses PET imaging alongside MRI and SPECT, which together are powerful techniques to help us understand disease mechanisms and investigate biomarkers for the early identification of disease and to track disease progression.
Our group has really pioneered methods that enable us to actually profile patterns of gene expression and gene regulation in those individual cell types. And this allows us for the first time to really get at the molecular underpinnings of disease taking place in specific cell types in the brain.
Using the stem cell models that we have developed, we have discovered that certain kinds of molecules within a cell called non-coding RNAs are dysregulated, and by restoring the levels of these molecules, we have found that we can enhance the survival of the neurons.
And because we're a medical school and our research is really firmly rooted in a hospital setting, we're able to really rapidly translate some of our very basic neuroscience findings into ideas of developing new treatments and drugs and therapies that might ultimately help individuals suffering from these very debilitating diseases.